story that starts with a clinical problem we have. It's very different from what convince you how important Chemaxon is into our project. But we're going to start with a clinical issue and work backwards to the Harvard Medical School is very strict disclosure policies. I'll just say that I'm going to discuss the off-label use of a particular drug and the FLAIR consistent with investigational use only. Everything I'll show you with FLAIR is owned by my institution. I think someday we'll see royalties. I'm the founder and I'm the director of a office that is trying to promote this technology. So it's a really simple question, one would think. Let me turn the mic down a little bit. Does this man, woman, or child have cancer? Clinical question that really impacts everyone in this room, either personally or your family and friends. And if so, how much, where is it? So cure it. And unfortunately, right now, we don't have an answer to that question as oncologists, trained as oncologists, or something I've seen in the clinic. And the problem really is one detection. We have the ability right now to detect about 1 billion cancer cells in the body. And to give you some perspective, cancer starts off as a single cell, double. Eventually, we reach the billion cell mark. Billion cells are about the size of a fungi. Deposition occurs at about 1,000 billion, a trillion cells, kilograms. That's the maximum in the body understanding. So we have a detection problem. We're detecting cancer here at 1 billion. We go to patients 10 cell divisions, or 10 net cell divisions. And we then have to invent this word called remission. Remission is really good fall below the detection threshold. Now, how far do we have to go in oncology to make a big impact? If we get to 1 million cells, we're going to have a huge impact on things. So that's right around the inclusion switch, and that's where things are going to change. But we're not there. We're 3 orders of magnitude away. How do we get there? So we resigned ourselves over a decade ago to plug-and-play type of chemistry. So for chemists in the room, I apologize. Very simple chemistry, the NHS aspect in front of your knees, to weld together a homing device, small molecule targeting ligands, with some type of effector domain, either a light bulb in the case of diagnostic agents, that I'll show you the department, or in the case of a cytotoxic agent, a bomb. So we're really, we spent over a decade optimizing this part, optimizing every part of the chemistry there. So we're really done with that. I think the paper is very interested. What right now the focus of the lab for the next decade is this homing device. These small molecules to target tumors over normal cells. It doesn't have to be absolute targeting. It just has to be good enough. It has to be relatively good, such that we can achieve or get towards our goal 3 laws of detectability that we now lack. So to do this, we've been developing, this is a work in progress, a system, a robotic chemistry system that uses 6 custom robots linked together through JChem and the software infrastructure to automate every part of the discovery and screening process. And we decided to go with coderless on a combinatorial library for a variety of reasons. And I think what's unique about this is, number one, it really starts with a clinical problem, works backwards with chemistry. But secondly, we've made a lot of effort to make it a circle so that in a completely automated fashion, we can not only identify the compound, but resynthesize it in a way. So I'll take you through these robots and show you how JChem is used to link everything together. And JChem really is the glue that's what's used together. This is just what the room looks like you're looking at it from the top of the corner of the ceiling. There's 6 robots scattered here. We'll go through each of them, and through Ethernet, everything is connected to the central workstation. And a very small number of people, therefore, can run this infrastructure. So we'll start just with the software itself, not one of the formal 6 stations, but really, again, the glue. And that's through Chemaxon's JChem. This is where we perform in silico enumeration. We store our databases. We do any pharmacophore modeling that's necessary and have our 2D and 3D viewing. We've linked that up to an Oracle database that feeds into the hospital's petabyte storage system. And then we have a variety of custom C++ and C# routines, all with using iLink Pro. We're Calibre-based for our liquid handling, all linked together. And I'm going to take you through this and show you how we do it. So the first station, station 1, is for automated split pool synthesis. And when we started this, we thought it'd be a lot easier than it was, but it turned out to be a real bear.
because the human body can pipette beads quite easily, but to get robots to do it is hard. So we have some custom devices here to manipulate 500 micron beads very efficiently that can aspirate and dispense very precisely and handle the liquid. We have microwave integrated, conventional heating if needed. So this particular station can synthesize compounds, synthesize libraries in the 5,000 to 10,000 compound range. But what we use JChem for is to make this fully integrated. So GUI-based construction of the libraries, we're going to free our chemists from anything that other than just clicking and dragging to create these libraries. We use in silico enumeration that I'll show you a little later to produce not only final products but stop products along the way, because that's really the key for the encoderless detectability, is to be able to take these products of the in silico enumeration using reactor and a custom NIME node I'll show you, and feed it into the mass spec so we can perform automated analysis. Station 2 is simply a sorter, a bead sorter, taking these reactions that have 5,000 or 10,000 beads and putting one bead in a single well of a 3D4 well plate. Something a human can do, although it's tedious, but to get a robot to do it was actually quite hard. And we've been working with industry, a company called Tech Elon, and we've solved the problem, and it's really, really quite nice. Fully automated sorting of any unlimited number of beads into an unlimited number of 384 well or 96 well plates, and we've also integrated quality control so that we can tell whether one bead or two beads, even though it's a relatively rare event, we want to discard anything with two beads in a well. Station 3 is simply liquid handling, cleaving the compounds from the beads, getting them into individual wells without the beads around. And we do this several different chemical ways. One of our favorites is UV cleavage, and we've worked with Caliper to develop a custom UV cleavage box that works really well, letting us, as you see here, cleave this fluorescent compound from the bead into the well, and then master-slave plate. Station 4 is one of the keys. We worked a long time on this, being able to imprint glass slides, but do it in completely aprotic conditions, do it reliably, and to simultaneously be able to create 5,076 spotted MALDI slides. Again, the key to the encoderless detection is being able to feed these data into MALDI, and we can now do 5,076 compounds in an automated fashion that mimic our glass slide. Now, why do we want the compounds here, under aprotic conditions? It's for the live cell screening, Robot 5. This is a microscope that automates the process, but what we're able to do is take living cancer cells, I'll show you here, and just pan them over the slide. And in serum, in warm serum, in medium, in the same context in which they're seeing ligands in the body. This is just to show you how we do it. We load the cells with various near-infrared fluorophores. I won't go into the technical details, but there's a reason for that. We're able to do 2 or 3 channels, depending on what we're trying to achieve, of live cell screening. And every spot here is a 350 micron chemical spot put down by Robot 4. And every tiny little red or green spot is a cell. These are the living cancer cells that have been screened. Now, this particular spot here is just a control. It's polyalamine. It'll bind every cell known to man. This gives us a nice quality control for making sure the system works and getting our counting algorithms worked out, et cetera. And because there's no specificity, these are 2 different cancer cell types, actually 2 different phenotypes of prostate cancer that differ only in a few cell surface receptors. We're able to see both, of course, because this is non-specific, and we see the readout. If you do the same experiment with ligands we've developed over the years using conventional chemistry, and again, for the historical sake, the transition from this 80 nanomolar to 700 picomolar binder took us 7 years, and I think it was 5 postdocs and a couple $1 million. We don't want to do that anymore. We need to automate it, which was the push for this whole system. But the system works quite well. Here's a cancer cell that binds this particular ligand, a cancer cell that doesn't, and you can fully automate the screening process now. So, and then I'll just end with Station 6, which is the encoderless detection of what was initially enumerated. We don't want to spend a lot of time or energy tagging. We've gone through the literature, and we can see so many counterexamples of when tagging fails rather than when it works. We want to eliminate that. And again, by using JChem's in silico enumeration and this, the MALDI technology we've developed, this was in collaboration with a company called HST in Korea, this custom slide that's really just unbelievable. We're able to fully automate the MALDI screening process. And the system is calibrated from 121 to 1436 Dalton, so we can encompass most of the chemical sprays people are interested in this room. And to make it work, we had to build a custom NIME node that really works well. It interfaces with the GUI and is able to take the in silico enumerated stop products and final products and do pattern recognition with the MALDI data. This is just an example of this. So here in a reaction where we had 5 series reactions with 2 scaffolds, 4 reactants each. So it's 5,000 compound. It's our typical library. We're not striving for more than that. We can enumerate this library now in 6 minutes, and we're still customizing it. And that's giving us the data set. It, of course, takes 24 hours to accumulate the data in MALDI, but at least the enumeration is working incredibly fast. And this is a summary of the whole system right now by station. So in terms of synthesis, we're really only limited by bead volume, but we're not going to be able to do more than 10 to the 5th beads. We're going to be happy at 10 to the 4th. Being able to take the beads from bulk solution and put them into individual wells of a plate isn't really limited. It's just limited by the number of twisted tube robots we happen to have right now. So it's at about 30,000 compounds. Master-slave plating is just a matter of time and consumable costs. Being able to plate the slides is somewhat rate limiting, although we're getting it faster and faster. Screening the slides isn't really an issue anymore. The microscope can now do 280,000 compounds, and then the MALDI is 5,000 compounds every 13 hours, fully optimized. So roughly our goal, and we're not there yet, but we're very close, is 10,000 compound libraries every 2 days. Again, freeing up our chemists to think about where we should be diving into chemistry space for particular tumors and letting the system do most of the work. And what's important to us, because again, we're approaching this from the clinical problem, we've designed this system to screen biopsies, and we're now capable of 100 cancer biopsies against 5,000 new compounds every 2 hours, or taken a different way, 1 biopsy against 100,000 compounds. But it's very rapid screening with small numbers of cells, and that's the only way to make it work in the clinic. Any system that's going to require more than a few thousand cells just isn't going to fly clinically, because we only have small numbers of cells available for medical legal reasons. Most of it goes to pathology and is inaccessible. And if we were looking to the future, why are we doing all this? Well, we're doing this because right now we're failing in treating cancer. Everyone in this room knows that. 50% of the patients who are going to be diagnosed this year of cancer are going to die of their cancer. So we're failing. And we haven't made much progress, even though the news says we have. We really haven't made it in the big volume diseases. And so where do we need to go? Well, now that we're getting closer and closer to phenotypic recognition of the differences among cancers, and breast cancer, for instance, now can be pretty neatly divided into only 4 phenotypes. And that's an important advance. We're not really harnessing that advance yet. So the dream we have, the vision we have, is over the next decade accumulating hits on these chemical libraries. It'll be very slowly, but slowly accumulating to the point where on one of these chips that'll hold 5,000 compounds, we have compounds that are breast cancer specific, or rather relatively breast cancer specific. It only has to be specific to cancer over normal elements of breast, connective tissue, and adipose, et cetera, that we're actually pretty good at differentiating. Same with prostate cancer. And now why does this matter? It matters because a decade from now, when a patient walks in with breast cancer, we'll be able to take a small number of cells from a biopsy and have screen it on this chip very rapidly and find the combination of agents that are going to attack the majority, if not all, of the cells in that tumor. And I didn't mention it, but we actually have 5 channels of fluorescence on our system, so we can phenotype at the cellular individual cells for binding to individual compounds. So we can really do quite a nice analysis on a very small number of cells from a patient. Well, this patient may have this combination. The next patient may have a completely different combination. Now, I'm not pretending that we have the regulatory structure to be able to do this. If you told me right now you have 5,000 compounds in the freezer that can cure cancer, there's no way the FDA could handle that. We're still in a one compound mode, but we're all hopeful that'll change over the next decade or two as advances are made. So we have nothing to fight over right now, but I can tell you the fight's going to be a decade from now is how to get combinatorial treatment into the clinic without hitting a massive regulatory roadblock. But that's a problem for the future. And so who are we? You don't know me from Adam. We're coming again from the chemistry side of this, but we're the Center for Molecular Imaging at Beth Israel Deaconess of Harvard Medical School. We have 6 laboratories working day and night on coming up with new technology for seeing cancer in smallest possible state. We have a state-of-the-art small animal imaging facility if anyone needs small animal imaging work. And we're in the process, through a stimulus grant, building a translational imaging facility where these compounds can get into patients very quickly, not as combinations yet, but as individual molecules. And I'll just briefly mention this because it was funded by a stimulus grant. Construction's occurring as we speak. We're just having all the permitting and design approvals. But in this facility, we'll be able to take compounds hooked up to certain types of light bulbs, mostly for positron emission tomography and for image-guided surgery, and move these molecules into the clinic very rapidly, on the order of months instead of years. And again, we have the track record, and we're already doing this without the facility, and hope to expedite it. And I just wanted to end with a real-life example of why this matters. Why does the chemistry matter? Why does JChem matter? And this is this FLAIR imaging system I mentioned. It's a very small system that uses invisible light that's in this room, but we can't see it. But near-infrared light has certain properties that are beneficial. It can see through blood. It can see through tissue several millimeters. Not centimeters. It's not X-ray types of energy, but it can go millimeters. And in cancer surgery, millimeters matter a lot. And I'll just end with a real-life example. This is a woman undergoing breast cancer resection. And we're using this system in conjunction with these special light bulbs to see sentinel lymph nodes and being able to tell the surgeon exactly where the node is and give them the ability to resect it under image guidance. And as this JChem-based system moves towards the clinic, we hope to be doing this with the actual targeting the tumor specifically, both in breast and prostate cancer. But it's, the future is close. We're no longer decades away. We're actually getting closer. And we're very grateful to Chemaxon for giving us the tools to move this project forward so quickly, because we couldn't have done it without it. I wanted to thank my group that does this, run by Hak-Soo Choi, a brilliant PhD scientist, expert fluorophore chemist, as well as leader. And each area has really superb people on it. Jung-Han Lee, who's here in the audience, handles all the MALDI. We have an excellent engineer keeping everything going. And JChem Oracle integration was quite a big project, as you can imagine. We're grateful to Rafi Tejas, Gabe, and Jung So for doing that, and Khalid Nasser, one of our chemists who's working closely with the group. We have various websites that you're always willing to visit, and there's a variety of reviews if you're interested in any aspect of imaging cancer and why this matters. And thank you, and I'll be glad to answer any questions.
Thank you, John.
Yeah, that's a great question. So the question, for those of you who didn't hear it, is, where do we go from here if we have hits? What are we going to do? Well, frankly, we're not there yet. Right now we're so enveloped. Everybody in the group is working to just finish the loop and get all of these details worked out. But that's one of the reasons we went with JChem, for everything I've heard at this talk, and these talks have been fantastic. I have a much better sense for what the capabilities are. It's almost unlimited. So no, we haven't addressed that yet. I think that's going to be done with collaboration with industry and with people a lot brighter than we are in the chemical domain. But we chose JChem for that reason, so it would be enabled.
Hi, Dana Vanderwall, Bristol Myers Squibb. Yeah. Have you thought about salting in existing agents to your screening library?
Well, we're doing that now just to optimize. So we're going to be using existing agents just to make sure the concept works. And how good is it? We haven't closed the loop yet. We have the enumeration done. We have the MALDI done. We haven't done the pattern recognition. So how good are we? We don't know yet. Maybe we'll be able to report that back in a year or two.